Hello, I'm Professor Sims, and in this video, I'll discuss DNA structure and function. This is the fourth in the series of 10 lessons held as part of my General Genetics course. If you are a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this unit include describing key experiments that helped identify DNA as the genetic material, explaining structures of DNA and the Sanger method of sequencing DNA, identifying similarities and differences in eukaryotes versus prokaryote DNA and DNA replication, explaining the process of replication in both prokaryotes and eukaryotes, and the enzymes that are involved in both of those. And then we'll talk about the different types of mutations that can happen during DNA replication and how DNA repair mechanisms help to compensate for those mutations. Our understanding of DNA began with the discovery of nucleic acids and the development of the double helix model. In the 1860s, Friedrich Miescher, a physician, he isolated phosphate-rich chemicals from white blood cells, which he called nuclein. He actually dug dirty dressings out of the garbage and collected it, and he recognized later on that the nuclein was DNA that he was able to extract from the cell nuclei. In 1928, a British bacteriologist, Frederick Griffith, conducted groundbreaking experiments on bacterial transformation using Streptococcus pneumoniae. He worked with two strains of the bacterium called rough and smooth, R for rough, S for smooth, which differed in their appearance on nutrient agar plates. Griffith observed that live S strain caused pneumonia and death in mice, while live R strain was non-pathogenic. He conducted an experiment where he injected a mixture of live R and heat-killed S strain into a mouse, and they died. The mice died. And then he isolated only one live S strain from their bodies. And he concluded that something from the heat-killed S strain transformed the live R strain into a pathogenic form. This transforming principle laid the foundation for further research. Scientists Oswald Avery, Colin McLeod, and Macklin McCarty. In 1944, they were trying to identify the transforming principle. They had isolated proteins and nucleic acids from the dead mice infected with the S strain. And by using enzymes to degrade each component separately and then testing their ability to transform the R strain, they discovered that only the DNA degraded mixture lost the transforming ability. And this led them to conclude that DNA was the molecule responsible for transformation. Despite that finding, DNA was considered too simple to carry all of the information that it carries compared to proteins, right? Proteins, with their diverse amino acids, they were thought to be more likely to be the genetic material. An experiment conducted by Martha Chase and Alfred Hershey in 1952 proved that DNA was indeed the genetic material. They studied a bacteriophage, which is a virus that infects bacteria. And they labeled it with phosphorus 32P to mark it, right? By observing which radioactive molecule entered the bacterial cells after infection, they were able to find that only the phage DNA entered and carried the information to produce more phage particles, right? So this experiment solidified DNA status as the genetic material. And this is figure 14.4, and it's showing that in the experiments I was just talking about, Hershey and Chase, the bacteria were infected with a phage that was labeled 35S or 32P, and they found that only the 32P was able to enter the bacterial cells. 32P was the DNA, whereas the 35S here, this is the protein coat. Around the same time that these experiments were happening, a biochemist named Erwin Schargraf made significant discoveries about DNA. He found that the amounts of adenine, thymine, guanine, and cytosine varied among different species, but they remained consistent within the tissues of the same individual or between individuals of the same species. Schargraf observed that the amount of adenine always equaled the amount of thymine, and the amount of cytosine adenine always equaled the amount of guanine. So there were equal proportions of purines, the adenine and guanine are purines, and equal proportions of pyrimidines, the thymine, cytosine are pyrimidines. And this observation became known as Chargaff's rules, and they were crucial for Watson and Crick when they later discovered the DNA molecule. And we'll see some more in a bit about how the purines and pyrimidines fit together in the helix. And then also here we have uracil. Uracil is in RNA, but not in DNA. And then thymine is in DNA, but not in RNA. So DNA is made up of nucleotides, and a nucleotide is made up of parts too, so we're going to talk about those. A nucleotide consists of a nitrogenous base, a 5-carbon sugar, and a phosphate group. The nitrogenous base can be either a purine, such as adenine and guanine, or a pyrimidine, such as cytosine and thymine. Purines are the ones that have the double ring structure, and pyrimidines only have one ring. This sugar that is in DNA is called deoxyribose. That's how it gets its name, deoxyribonucleic acid. The sugar that is in RNA, it's called ribose, which is why RNA stands for ribonucleic acid. The nucleotides are all linked together by phosphodiester bonds, where the phosphate of one nucleotide is attached to the sugar of the next nucleotide. And this creates a chain with a 5' phosphate and a free 3' OH. Here's what that looks like here. This is the 5-carbon sugar and the phosphate group attached to it, and then those attached to either one of these pyrimidines or a purine, and all of those parts attached to each other is what is called a nucleotide. In the 1950s, Francis Crick and James Watson collaborated to determine the structure of DNA. They were inspired by the work of scientists like Linus Pauling and Maurice Wilkins, who were also investigating the DNA structure. They ended up utilizing X-ray diffraction pattern that was discovered by Rosalind Franklin. She had been working in an X-ray diffraction lab, and she had lots of data and was actually able to photograph the double helix. And Watson and Crick were obviously inspired by that work, and they combined their expertise in X-ray diffraction analysis, and they proposed that DNA is indeed a double helix consisting of 2 intertwined strands. So up until that point, some people thought that it was just one strand, some people thought that there was like a triple helix thing happening, but now it was confirmed that it is a double helix. The 2 strands are anti-parallel, and then the sugar phosphate backbones form the outer structure, while the nitrogenous bases stack inward like rungs on a ladder. And then the uniform diameter of the helix, so the width of the helix, stays the same, and that's because of the complementary base pairing, the A to T, C to G. So let's look at that in a bit more detail. This is figure 14.7, and it's got the double helix structure here on the left. B here is a close-up showing the phosphodiester bonds, the hydrogen bonds in the middle, phosphodiester bonds on the outside. And notice that it's only purine and pyrimidine, and that's how it stays the same width in the middle. And that's why they only bind to those bases and to no other bases. A only goes with T and C only goes with G. And then C over here is showing the major and minor groove. The grooves work as binding sites for DNA binding proteins that work during transcription and replication.
Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has 3 components: the 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in DNA, deoxyribonucleic acid, are called deoxynucleotides. Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. Thus, we can see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand, 5' to 3'. Looking within the sugars, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left, and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3', and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that this double-stranded DNA is still a double helix, and we have simplified the representation by flattening and unwinding the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through 2 hydrogen bonds, and cytosine preferentially pairs with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT, or TA, and GC, or CG, base pairs is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angles to which the base is attached to the backbone. Other base pairs, like GT for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs.
DNA sequencing was a really lengthy and expensive process up until the 1990s. Around that time, advancements in technology and automation made it safer, cheaper, and faster to sequence DNA. Fred Sanger is the creator of the dideoxy chain termination method, and we usually just call that Sanger sequencing. The method involves using dideoxynucleotides, DD-NTPs, in addition to the regular free deoxynucleotides, the D-NTPs. You use the dideoxynucleotides that have a 3' OH group that's missing, and you use those as chain terminators. So by adding a specific ratio of the dideoxynucleotides to the regular nucleotides, you have different sized DNA fragments that are generated. The DNA sample is denatured, it's divided into separate tubes with primers and nucleotides, and DD-NTPs are added to each tube, and then each dideoxynucleotide has a fluorescently labeled tag. So it's got a fluorescent label for detection, and you have chain elongation stopping once the fluorescent tag is incorporated, and results in fragments that are separated by electrophoresis, and a laser scanner comes and reads the sequence by detecting the fluorescent marker. In gel electrophoresis is a technique that's used for separating DNA fragments by their size and by their charge. What you have is a gel that's made out of agarose. You have agarose is a polysaccharide polymer that comes from seaweed. It's a solidifying agent. So you mix the agarose powder with a buffer, you heat it up, you pour the gel solution into a casting tray, and you allow that to cool and solidify. And once it's solid, you take the DNA samples and you put them into little wells that have been cut out of the gel. And since DNA has a negative charge, if you put an electric current across the gel and you put the positive charge at the opposite side of the gel, the DNA wants to migrate through the gel to the positive end, and it separates the DNA based on the fragment sizes. So the smaller fragments go a lot further, a lot faster than the larger fragments because they're physically moving through a semi-solid gel. So the smallest fragments end up the furthest away from where they started, and after separation the gel is stained with a DNA-specific dye so that you can see it. You can visibly see the DNA in the gel. So figure 14.8 is talking about Sanger sequencing. This can be kind of confusing, but if you understand how PCR works, you understand how sequencing works. Okay, it's very similar. What you have is you have a reaction mixture that has your DNA, and it's got the enzymes in there to make the reaction go, and then it has the free nucleotides, your adenine, thymine, guanine, cytosine. You add them in there, and in the PCR, what you're doing is you're amplifying the DNA. You run it through hot and cold cycles, and the DNA denatures, and then it elongates, it replicates. The same thing is happening in Sanger sequencing, except in addition to everything you would use for PCR, you also have these little things that are altered artificially. They've had the 3' OH group removed. So when that OH group is gone, the next nucleotide that wants to add to the growing DNA chain, it can't because it doesn't have that little OH group to hook onto. So if you have these dideoxynucleotides in your reaction mixture, they're going to randomly bind to DNA that's replicating. And when they do, it's going to stop the replication because there's nothing, it doesn't have that OH group to hook onto. So it stops the reaction. And then if you put in place of that OH a fluorescent molecule, it works as a little flag, like a little tag that says, "Hi, here I am." And in that way, you can actually see the position. If you have dideoxy, adenine, guanine, they have color coding. So once you run it through the gel, you can see an image that the laser has put out, and it tells you what color it saw, it tells you what position it saw that color, and the peaks tell you how confident it is. So like there is some background noise down here, but these are very big peaks, so you know that this is good data. This is definitely a G. This is definitely a G. So that's how Sanger sequencing works. And then this is the gel electrophoresis, what it looks like. So you put it in these wells, you put the DNA in these wells, and then after this is loaded, she put a cover on it and turn on a power source that runs electricity through the gel. And then after it's done running and you stain the gel, you can put this under a light and see the DNA that's been separated by side. So let's move on to DNA packaging in cells. This is a good time for a reminder about the differences between eukaryotic DNA and prokaryotic DNA. Prokaryotes have a simpler structure compared to eukaryotes. They're typically only going to have a single circular chromosome that's located in the nucleoid area, right? Because they don't have nuclei. In order to fit such a big genome into a small bacterial cell, the DNA undergoes supercoiling. Supercoiling is where the DNA is twisting either in an underwound or overwound. It's twisted up. It's not relaxed, right? So proteins like DNA gyrase play a role in maintaining this supercoiled structure. Eukaryotes have a nucleus. They have a nucleolus, and they have chromatins. They have much more complicated structures, and they're much larger than prokaryotes. They have a linear chromosome that uses a different packing strategy within the nucleus. At the fundamental level, the DNA is getting wrapped around a protein called histone, and it forms what is called a nucleosome. Histones are conserved proteins that are rich in amino acids, and they combine to make nucleosomes. The nucleosomes kind of look like a bead on a string, and it has the DNA tightly wrapped around it. And then it becomes connected to adjacent nucleosomes by linker DNA. And then here comes another histone that makes this fiber around the chromatin. During metaphase, the chromosomes get even more condensed with the aid of scaffolding proteins, and at this point it's about 700 nanometers in width. During interphase, the eukaryotic chromosomes exhibit two distinct regions. One is heterochromatin and one is euchromatin. Heterochromatin is densely packed and it contains non-expressed genes. It's found near the centromere and the telomere, so we're talking here and out here. The euchromatin is less dense, but it contains actively transcribed genes, and it's packaged around the nucleosomes, but it's not as tightly compacted. The discovery of the DNA helix structure actually hinted at the mechanism of DNA replication. Watson and Crick wrote this groundbreaking paper in 1953, suggesting that the specific base pairing in DNA allows for accurate copying of genetic material. According to their model, the two strands of the double helix separate during replication, and each strand serves as a template for the synthesis of a new strand. However, the precise details of how this would work were unclear at the time, and they had three proposed models. One was conservative, the other was semi-conservative, and the last one was dispersive. The conservative model suggests that parental DNA remains intact, while the semi-conservative model proposes that each parental DNA strand acts as a template, resulting in one old and one new strand in each of the newly formed DNA. The dispersive model suggested that both copies of DNA have segments of parental and newly synthesized DNA interspersed. Here's an illustration of the three models that had been proposed: conservative, semi-conservative, and dispersive. You may already know which of these models ended up being correct, but we will go into that in a moment. Messelson and Stahl in 1958 did experiments using E. coli to investigate DNA replication. So what they did is they grew E. coli on a medium that contained a heavy isotope of nitrogen, that's 15N, and then they transferred it to a medium with a normal isotope, the 14N. By isolating and centrifuging the DNA, they observed bands that indicated intermediate positions between 15 and 14N, and this ultimately confirmed that the semi-conservative model was the correct one. So this is figure 1413 showing that experiment. Over here, the red is the heavier isotope of nitrogen, and then this one is the normal, the regular isotope. And essentially what they did was a freeze-thaw method of DNA extraction. So they just got the cells to burst open and release the DNA, and they would have that process go on in the heavier isotope of nitrogen, and then they would remove the DNA from there and put it in the 14 nitrogen. And they found that in the F1 generation, the first round of offspring, it didn't just have 15, it didn't just have 14, it didn't have something that was completely different that just had pieces of them interspersed. It had like a 50-50 ratio of DNA from here and here. So they were able to see that the parental DNA was intact, but it was one strand of it. And then there was another strand that came from the other parent. In this case, it's a test tube, but you get what I'm saying. During DNA replication, each of the two strands that make up the double helix serves as a template from which new strands are copied. The new strands will be complementary to the parent strands, and when two daughter DNA copies are formed, they have the same sequence and they are divided equally into daughter cells. When DNA replicates, the double helix unwinds, and then each strand serves as a template for the synthesis of a new complementary strand. This ensures that the daughter DNA have the same sequence, they're distributed equally. In prokaryotes like E. coli, DNA replication occurs rapidly and with high accuracy. We should go ahead and introduce the key enzymes in prokaryotic DNA replication, as these enzymes play a crucial role in adding new nucleotides to the growing DNA strand. Nucleoside triphosphates provide both the nucleotides and the energy that's needed for polymerization. Prokaryotes have three main polymerases. DNA polymerase 3 is the one that is responsible for DNA synthesis, but they also have DNA polymerase 1 and 2. DNA replication in prokaryotes begins at the origin of replication, which is a specific site on the circular DNA that is found in prokaryote cells. Enzymes called helicases unwind and separate the double-stranded DNA, and this is done with the aid of ATP hydrolysis, and it creates what is called a replication fork. Single-stranded binding proteins prevent the single strands from reforming a double helix. The enzyme DNA polymerase synthesizes the new DNA strands, one of each template strand, and it uses free nucleotides present in the cell. The process is bidirectional, with replication proceeding in opposite directions from the origin until the entire circular DNA molecule has been copied. DNA polymerase has two important limitations. It can only add nucleotides in the 5' to 3' direction, and it requires a free 8' OH group to form a phosphodiester bond with the next nucleotide. So this means that it needs a primer with a free 3' OH end to initiate DNA synthesis. RNA primase synthesizes a short RNA segment called the primer that's complementary to the template DNA. And then DNA polymerase can then extend this RNA primer by adding nucleotides one by one. I'm aware that I guess I was missing a figure there, but that's okay. We'll have a video in just a moment where you can see all of this happening. During replication, the replication fork is moving really fast. In prokaryotes, it's at about 1,000 nucleotides a second. Topoisomerase prevents the overwinding of DNA ahead of the fork, so it creates temporary nicks and it reseals the DNA. At the replication fork, there's a challenge there because the two template DNA strands have opposing orientations. The leading strand, which is complementary to the 3' parental DNA, can be synthesized continuously towards the fork. However, the lagging strand, complementary to the 5' is synthesized away from the fork in short little fragments that are called Okazaki fragments. Each Okazaki fragment requires a new primer to initiate synthesis, and a sliding clamp protein provides the DNA polymerase a place to bind to, and it keeps it there while it's adding nucleotides. The leading strand extends from a single primer, while the lagging strand requires multiple primers for each Okazaki fragment. As synthesis progresses in both directions, the RNA primers are replaced with DNA, and the gaps left by the removed RNA are filled by DNA nucleotides using the exonuclease activity of DNA polymerase 1. DNA ligase seals the remaining gaps, the nicks between the newly synthesized fragments, by forming phosphodiester linkages. After replication is complete, the two DNA copies separate and move into different cells during cell division. So yes, we need to visualize this. So let's have a look at this video here.
DNA replication is the process by which a duplicate copy of the genetic material is made before a cell divides. The steps in replication are highly conserved in all organisms. There are a few details that differ between bacteria and eukaryotes, but we focus on bacterial replication in this animation for simplicity. The process of DNA replication is broadly described as having 3 phases: initiation, elongation, and termination. Let's begin by considering the initiation phase. For copying to take place, the double helical structure of the DNA must first be opened at a site termed the origin of replication to allow access by the machinery that will copy the DNA. This opening is regulated by a series of initiation regulatory proteins not shown here. Once open, a DNA helicase, a ring of 6 subunits, is loaded onto one strand of the DNA at each side of the replication bubble. This establishes 2 replication forks that will move away from each other as replication proceeds. The synthesis of DNA is catalyzed by an enzyme called DNA polymerase. However, DNA polymerases cannot start a new strand from scratch. Instead, they can only elongate an existing polymer of DNA or RNA. Consequently, a primer sequence needs to be made from which DNA polymerase can continue its synthesis. Primers are made by an enzyme called primase. Once access to the single strands of the DNA has been obtained, the primase is loaded onto the DNA. The primase synthesizes a small stretch of RNA by copying the sequence of nucleotides on the template strand. A DNA polymerase will later elongate this small RNA stretch. The next steps allow loading of the DNA polymerases onto the DNA to copy the entire chromosome. The first component that is loaded is the sliding clamp, a ring-shaped structure that binds at the 3' end of the newly made RNA primer. To get onto DNA and encircle it, the clamp is loaded by a clamp loader complex. The 5-protein clamp loader complex binds to the sliding clamp, and binding of ATP allows opening of the sliding clamp ring. The opened ring then encircles the template primer junction at the 3' end of the primer. Interaction with the 3' end of the DNA stimulates ATP hydrolysis, and the clamp loader dissociates from the DNA to be replaced by the replicative DNA polymerase. The 3' end of the primer is positioned in the polymerase active site to allow addition of nucleotides to this end. During elongation, the replication bubble gets bigger as the helicases move apart, establishing 2 replication forks that move in opposite directions. The sliding clamp and polymerase unit at each replication fork moves along the DNA as the helicase unwinds the double-stranded DNA to expose single strands. Let's focus on just the right-hand fork for the moment, as the processes at both forks are identical. Polymerases are loaded onto both the top and the bottom DNA strands at the fork. Because polymerases only synthesize DNA in the 5' to 3' direction, the two strands must be copied in opposite directions. The bottom strand in this fork is the leading strand. The polymerase on this strand moves continuously from left to right, synthesizing DNA as it travels. The top strand is the lagging strand, on which just a short stretch of DNA is made by the polymerase elongating in the 5' to 3' direction. After synthesizing this short stretch of DNA, the polymerase then dissociates and a new polymerase binds at the fork to elongate the next RNA primer. So priming and elongation are happening repeatedly on the lagging strand as the replication fork moves along the DNA. Synthesis is discontinuous. The short stretches of DNA that are made on the lagging strand are called Okazaki fragments after their discoverer. During the course of replication, these fragments are stitched together. When the lagging strand polymerase runs up against a previously made Okazaki fragment while in the process of synthesizing DNA, the replicative DNA polymerase 3 is replaced by DNA polymerase 1, which has 5' to 3' exonuclease activity. This exonuclease then degrades the RNA primer in front of it, synthesizing DNA as it goes. Once the RNA primer is removed, the two sections of DNA are joined by DNA ligase. Now the lagging strand has been elongated by one more Okazaki fragment. While this view of the replication fork is easiest to understand, in reality the leading and lagging polymerases are coupled and move in concert with the helicase. This coupling requires one DNA strand to be looped around so that the lagging polymerase can synthesize DNA in the 5' to 3' direction while moving with the fork. We explore this coupling in more detail in a separate animation. Much less is known about the details of the termination of DNA replication. Once the entire chromosome is copied, the two replication forks meet each other and are dismantled. The ends of the new DNA strands are then joined, again by DNA ligase. The two daughter DNA molecules now consist of one parent strand of DNA shown in grey and one newly made strand shown in green. Thus, DNA replication is termed semi-conservative.
So, figure 1414 is showing some of what we saw in the video. This is topoisomerase that keeps the DNA from recoiling. The helicase opens up the DNA, separates the strands. We have single strand binding proteins that help keep the strands separated. And then we have primase that's making the RNA primer. Then we have DNA polymerase that comes and attaches to that RNA primer and then begins going down the template to add nucleotides in the 5' to 3' direction. And this is a figure that's just kind of showing the overall process. You have a parent cell, and this is in prokaryotes, right? So remember, they don't have a nucleus, they don't have chromosomes, there's only one origin of replication, one replication fork forms, and then DNA replication happens down both sides of the molecule until the DNA has been doubled, and then when cell division occurs, you have one copy that goes into one daughter, and then another copy goes into this other daughter cell. And that's what it looks like in prokaryotes. And table 14.1 is giving all of the enzymes that are involved in prokaryotic DNA replication that we went through earlier. DNA polymerase 1 removes the RNA primer, replaces it with DNA. Polymerase 3 adds the nucleotides. Helicase opens up the DNA strand. Ligase seals up the gaps that are left. Primase makes the RNA primers. Right, so this is a very good table to keep all of those enzymes straight. Now, let's talk about eukaryotic DNA replication. So, in eukaryotes, the replication process is more complex because eukaryotes, remember, they're bigger. Everything about them is bigger, including their genome. And they have multiple linear chromosomes. So, humans, for example, we're eukaryotes, and we have about 3 billion base pairs in each set of chromosomes. So, replication is occurring at multiple origins of replication, multiple replication forks. forks, right? Remember, prokaryotes only have one. Eukaryotes have multiples throughout the genome. The rate of replication is slower in eukaryotes compared to prokaryotes, but again, there's just a lot more going on. They also have a larger number of DNA polymerases. There's 5 major ones: alpha, beta, gamma, delta, and epsilon. So let's look at the steps. The steps of replication in eukaryotes are similar to those in prokaryotes. Before replication starts, the DNA needs to be accessible as a template. So eukaryotic DNA is bound to histone proteins, forming nucleosomes. And the histones must be removed and replaced during replication, which contributes to the slower replication rate. Chemical modifications can occur in the chromatin to facilitate DNA accessibility. At the replication origin, a pre-replicated complex is formed with initiator proteins, followed by the recruitment of helicase and other proteins. Now, the helicase unwinds the DNA helix and it forms replication forks at each origin. Supercoiling ahead of the forks is resolved by topoisomerases. Primers are created by primase, allowing DNA polymerases to initiate synthesis. Okay, so that's very similar to what we saw in prokaryotes. Now we have different DNA polymerases to deal with. So 3 major DNA polymerases, alpha, beta, and epsilon, are involved. DNA polymerase alpha adds short DNA fragments to the RNA primers. Polymerase epsilon synthesizes the leading strand continuously, and the lagging strand is synthesized by DNA polymerase delta. And then a sliding clamp protein called pCNA helps to hold the DNA polymerase in place as polymerase delta encounters the RNA primer on the lagging strand. It displaces it from the template. RNase H removes the displaced RNA, and then DNA nucleotides replace it. The Okazaki fragments in the lagging strand are joined, and then DNA ligase seals the remaining gaps. So let's have a look at table 14.2. It is directly comparing prokaryotic and eukaryotic replication. So again, there's only one origin in prokaryotes. There's multiple in eukaryotes. The rate of replication is much faster in prokaryotes. Eukaryotes have a ton of different DNA polymerases. We talked about 5 of them. Telomerase is present in eukaryotes. We're going to talk about that in just a moment. Right, and the bulk of what is different is going to be here. What's happening at RNA primer removal and elongation, and all of these polymerases here. I think that this is a good place for another video. We're going to look at a video that compares directly DNA replication in prokaryotes versus eukaryotes.
Now moving into the difference between the prokaryotic and eukaryotic replication. Difference number 1 is regarding the site of DNA replication. In prokaryotes, DNA replication occurs in the cytoplasm, whereas in eukaryotes, the site of DNA replication is the nucleus. Difference number 2, in prokaryotes, DNA replication occurs in connection with the cell division, or it is the first step in binary fission or budding. Whereas in the case of eukaryotes, it is a complex process in the cell cycle. DNA replication occurs during the synthesis phase, that is the S phase of the cell cycle. Difference number 3 is regarding the ORI. ORI is the origin of replication, or a specific sequence that is required to initiate DNA replication. In the case of prokaryotes, there is a single ORI per DNA molecule. In the case of eukaryotes, the genome is single circular DNA molecule, and it is very comparatively less amount of DNA compared to eukaryotic system, and replication of DNA occurs at a single point in prokaryotic DNA molecule. Whereas in the case of eukaryotes, the genome is very vast, 50 or more times larger than that of the prokaryotic system, so that there are number of ORIs. Many ORIs are present. You can see these are the origin of replications, where the DNA replication starts. Thousands of ORIs in each eukaryotic chromosome, and replication occurs at several points in all these ORIs simultaneously in order to replicate this large amount of DNA. Now the difference number 4, it is regarding the number of nucleotides in the ORI. In the case of prokaryotes, this ORI is formed of approximately 100 to 200 or more nucleotides, whereas in the case of eukaryotes, each ORI is formed of approximately 150 nucleotides. Difference number 5, it is regarding the formation of replication fork. In the case of prokaryotes, these are the replication fork that is formed as a result of unwinding of the DNA strand by the helicase enzyme. In the case of prokaryotes, there are two replication forks and a single replication bubble. This structure is called as a replication bubble. As the DNA replication is bidirectional, a bubble formation occurs. There are two replication forks and a single bubble in the case of prokaryotic DNA replication. Whereas in the case of eukaryotes, a number of replication forks, you can see right here, a number of ORI, so that a number of many replication forks are present, and also a number of bubbles are present in order to replicate simultaneously all these origin of replication regions. Now the difference number 6. It is regarding the number of replicon. A replicon is a part of ORI along with a part of the DNA that is to be replicated. In the case of prokaryotic system, there is a single replicon, that is, the entire DNA is replicated simultaneously on a single stretch. So prokaryotic chromosome has one replicon. Whereas in the case of eukaryotes, a number of replicon is present, and these replicon may not be replicating simultaneously at a time. Maybe a different replicon may be replicating at different time. Now the difference number 7, this is regarding the initiation. Initiation of DNA replication in prokaryotes is carried out by proteins like DNA A, B, etc. and many other proteins. Whereas in the case of eukaryotes, initiation of DNA replication is carried out by a multi-subunit protein complex. It is often called as origin recognition complex. A number of proteins are involved in order to initiate the DNA replication. Difference number 8, it is regarding the DNA polymerase. As we know, in a DNA replication, there are two strand synthesis. One is a leading strand, which is synthesized continuously, whereas the second one is a lagging strand, which is synthesized as short fragments called as Okazaki fragments. In prokaryotes, these leading strand and lagging strand both are synthesized by the enzyme DNA polymerase 3. Whereas in the case of eukaryotes, this leading strand is synthesized by DNA polymerase delta and lagging strand is synthesized by DNA polymerase alpha. Difference number 9, it is regarding the size of the Okazaki fragments. In the case of lagging strands, the DNA is synthesized as short stretches called as Okazaki fragments. And these Okazaki fragments, the size of these Okazaki fragments in prokaryotes is approximately 1000 to 2000 nucleotides, whereas in the case of eukaryotes, it is much shorter. It is 100 to 200 nucleotides long. Difference number 10. In the case of prokaryotic DNA replication, it's very fast, rapid compared to eukaryotic replication, 200 base pairs per second, as the genome size is very small and there is not much requirement of a fidelity. As it is a single-celled organism, a mutation may not affect much. Whereas in the case of eukaryotes, comparatively, it is a slow process, 100 nucleotides per second. But remember, it is a large genome, so that a number of ORI, origin of replication, or replicon is working together to synthesize all this DNA, to copy all this DNA. Difference number 11, it is regarding the termination of DNA replication. Now telomerase or there is a specific termination sites or ter sites, specific sequences where the DNA replication stops. As in the case of prokaryotes, it is circular DNA molecule. At the end, there will be a daughter strand and a parental strand that is interlocked, and this locking is unlocked by the enzyme topoisomerase 2. But in the case of eukaryotes, as the eukaryotic chromosome is linear, it involves synthesis of specialized structures called telomeres. At the end, there is a telomere. There is an end replication problem that is sorted out by an enzyme called as telomerase, and it is an elaborate process also.
Telomeres are the ends of the linear chromosomes in eukaryotes. Telomeres play a crucial role in replication. Since DNA polymerase can only add nucleotides in the 5' to 3' direction, the leading strand is synthesized continuously until the end of the chromosome. But the lagging strand is synthesized in short stretches with separate primers. So when the replication fork reaches the end of the chromosome, the primer on the lagging strand's 5' end cannot be replaced. As a result, the telomeres or telomeres, which are repetitive sequences, they remain unpaired and then they may shorten over time as the cells divide. Telomeres consist of repetitive sequences such as TTA, GGG, in humans, repeated hundreds of thousands of times. Okay, so they're just long repeats. And the sequences protect the genes from being lost during cell division. Telomerase is an enzyme that adds telomeres to the chromosome ends. The telomerase contains a catalytic part and an RNA template, and it attaches to the chromosome end. It adds DNA nucleotides complementary to the RNA template, elongating the 3' end of the lagging strand template. And once that's long enough, the DNA polymerase can add nucleotides to complete replication. And this ensures that the ends of the chromosomes are replicated. Telomerase is active in germ cells and adult stem cells, but not in adult somatic cells. Scientists named Elizabeth Blackburn and her colleague Carol Greider, and also Jack, I can't say that, Sostak. Anyway, they won a Nobel Prize for Medicine and Physiology in 2009 for their discovery of telomerase and its actions. But further research later confirmed the telomerase's presence in human cells, including HeLa cells from Henrietta Lacks. Additionally, telomerase is necessary for rapid cell proliferation in embryos. Figure 1415 is showing the ends of linear chromosomes being maintained by the action of the telomerase enzyme. So just like I said before, the telomerase adds essentially nonsense DNA, all of these repeats to the end of the chromosome, so that when the chromosome is being replicated, none of the important genetic info is lost. Telomere shortening occurs in cells that undergo division, as most somatic cells lack telomerase, right? Because adult cells don't have the telomerase. So this is what essentially aging is. So recent studies in mice demonstrated that telomerase can reverse age-related conditions, which shows a potential in regenerative medicine. Reactivating telomerase in telomerase-deficient mice extended the telomeres, reduced DNA damage, and reversed neurodegeneration, and also improved organ function in the mice. So telomere reactivation may hold promise for treating age-related diseases in humans one day. In cancer, abnormal cells divide uncontrollably and they accumulate mutations. Cancer cells often have shortened telomeres, and telomerase becomes active only after telomere shortening. If telomerase activity in cancer cells can be inhibited during therapy, it could potentially halt future cell division and control the disease. DNA replication is a pretty precise and accurate process, but errors can occur, like when DNA polymerase inserts the wrong base, for example. Mistakes like that happen all the time, and they can have serious consequences, including what we were just talking about, cancer. Mutations can also arise from DNA damage, either induced or spontaneous. Induced mutations result from exposure to chemicals or environmental agents. Spontaneous mutations occur naturally within the body. Mutations can affect a single base pair, resulting in substitution or silent mutations. Some mutations replace one amino acid with another, impacting protein function. Others generate stop codons. Stop codons terminate protein synthesis, and a mutation could be causing that to stop too early, right? Trinucleotide repeat expansions lead to repeated regions of the same amino acid. And you have insertions and deletions and translocations. They're all different types of mutations. We'll talk about them some more in a minute. Mutations in repair genes have been linked to various forms of cancer. Somatic cell mutations cause uncontrolled cell division, while germ cell mutations are passed on to the next generation. So examples of that are hemophilia and xeroderma pigmenosa. Repair mechanisms exist to fix these errors, but sometimes mistakes go uncorrected. And like we were saying, repair enzymes themselves can be defective or mutated. So figure 1419 is showing a mutation that is caused by exposure to UV light. So basically you have thymines that are forming dimers. And in a normal cell, these would normally be excised by nuclease and then replaced by DNA polymerase. So these are some of your repair mechanisms. And it's done by these enzymes, nuclease, DNA polymerase, and then ligase, of course, fills in the gaps. Xeroderma pigmentosa, this we were talking about, is an example of where mistakes aren't corrected because the enzymes involved in repair of the DNA have formed the dimers, and there's mutations that are happening because of UV exposure. So the dimers are not getting repaired. So it's distorting the DNA structure, and it can lead to skin cancer. Then this is how DNA mutations can lead to changes in the protein sequence. So we're going to talk more about proteins later in the semester, but essentially proteins are just long chains of amino acids. And then the amino acids are encoded for in the DNA. So it's a 3 base pair codon that encodes for specific amino acids to be put in a specific sequence, right? If you have a mutation that is just removing, or it's just adding something, but it's not changing the code, right? So this amino acid can be encoded by this codon, but also by this one. So, no harm, no foul. You just have one base pair that was changed, but it didn't change the code. So that is a silent mutation. A missense mutation, there's a change in the code and there is a consequence because CCC and ACC encode for different amino acids. Nonsense substitutes a stop codon. So this THG, this is a codon that is telling the replication mechanisms, okay, this is where we stop. So we're not making more DNA, we're not making more proteins, we're not making any more amino acids. So this nonsense point mutation can be a very big problem. And then frame shift mutations, these also can lead to severe issues because you have either an insertion or a deletion that shifts the whole reading frame entirely. So here the T and the A get deleted and it changes every amino acid after that because the entire reading frame has been shifted over by 2 spots. And now a codon only has 3 base pairs. So if you've shifted it over 2 base pairs, you've really kind of changed the entire order of the codons. And so you end up with completely different amino acids being formed and completely different proteins. Now, as we saw a minute ago with the thymine dimers, DNA polymerase does do some proofreading. It reads the newly added base pairs before adding the next one. And if the base is correct, the polymerase continues. But if it's not, the polymerase will cut the phosphodiester bond and remove that nucleotide. And it does this using this enzyme called exonuclease. And then if this doesn't do it, if this doesn't fix the problem, there is another method. It's called mismatch repair. So usually you have repair enzymes that are recognizing and removing mismatched nucleotide. And figure 1417 is showing how the DNA polymerase can break this phosphodiester bond and remove this base pair and replace it with the correct one. In mismatch repair, if there's an incorrectly added base and it's detected after replication is done, the mismatch repair proteins can detect the base and remove it from the newly synthesized strand, again with a nuclease. And then the gap is filled with DNA ligase, just like we saw before. I want to show you a video of how this works. Again, it's really kind of difficult to visualize it and try to remember all the enzyme names and stuff. So this is the last video for this lesson, I promise.
DNA mismatches occur during DNA replication. For simplicity, this animation will show DNA as a flat molecule. Before replication can begin, a helicase needs to unwind the DNA molecule. In E. coli, DNA polymerase III catalyzes the incorporation of new complementary bases to the new DNA strand. This is only the alpha and beta units of the DNA polymerase III enzyme. However, it is possible for DNA polymerase III to incorporate a non-complementary base to the new strand. This is known as a mismatch. A mismatch could result in a faulty protein being coded for, so it needs to be repaired. The first step in the mismatch repair pathway in E. coli is recognition of the mismatch by the mut-S homodimer. Mut-S recognizes the mismatch and binds to it. Then, the mut-L homodimer binds to mut-S, ready for a later step in the mismatch repair pathway. Meanwhile, up to a kilobase away, the mut-H protein recognizes a GATC site. Mutate is a weak endonuclease, which will create a backbone incision between the guanine and adenine residues on the newly synthesized strand when activated. Mutate is able to differentiate between the newly synthesized strand and the template strand by the lack of methylation markers on the newly synthesized strand. This is why the pathway is also known as methyl-directed mismatch repair. In order for mutate to become activated, it needs to bind with mutel. This causes the DNA to bend. The newly formed backbone incision allows an exonuclease to enter and begin excising nucleotides from the newly synthesized strand. The exonuclease excises the mismatched nucleotide and stops excising just beyond it. Now, DNA polymerase III can begin to resynthesize the new strand, inserting the correct complementary base where the mismatch used to be. The final step is for DNA ligase to ligate the backbone nick. The mismatch pathway is now completed, and all the newly synthesized DNA is complementary to the template strand.
Thank you guys for watching. Don't forget to do the reading, check the description for more videos related to these topics, and leave your questions for me in the comments below.